Hello everyone, in this session we are going to learn about amino acids, a very simple concept. We know this particular chemical, that is the proteins, which are nothing but building blocks of our life. Without proteins we cannot survive. For example, you take enzymes, or you take hormones, or you take antibodies, or you take different types of receptors for smell, taste, etc. All these things are nothing but proteins. And what is exactly the protein structure? Okay, the protein structure is very simple, and you could see these are nothing but the monomeric units. Okay, you could see the circles, and which are linked by the peptide bonds here. Okay, so this is peptide bond and this is monomeric units. And what is this monomeric unit? This is nothing but amino acids. See, many amino acids are linked by peptide bond to make a long chain, which is nothing but called as poly means many, because many monomeric units are linked by peptide bonds. Okay, so that makes a polypeptide chain. What is polypeptide chain? Polypeptide chain is nothing but protein. Correct? Okay. So now we need to understand what is exactly this structure. This is amino acid actually. Okay, so now this concept is all about amino acid. So we need to understand what is exactly amino acid. Okay, so we write that. So amino acids. See, these amino acids are very simple structure, which is nothing but organic compounds. So they are organic compounds. Okay, so C must be there, carbon. And if you look into the structure of this, particularly it looks like this. A C is there, and to this C you could see carboxylic group is attached, and here the amino group is also attached, and here you could see R group. Normally it varies. So based on the type of the R group which is present in this particular amino acid, we can classify the amino acid into different types. Okay, so here you could see hydrogen. Okay, so this is the basic structure of amino acids. Now we'll have to understand, if we change this R to something like this. So R group in this particular amino acid is now H. Okay, then it becomes the simple amino acid that is called as glycine. So this is glycine. I just have to give you a very interesting thing about glycine. Actually, this glycine is amphoteric in nature. Okay, so we got a very good keyword here, amphoteric nature. So what is exactly the meaning of amphoteric? Amphoteric is nothing but any amino acid which actually acts as acid at the same time as base. So this is amphoteric. So glycine is a very good example for amphoteric. So why this particular glycine is acid as well as base, that is amphoteric in nature? The reason is very simple. You see this COOH here, which actually donates the protons, and you see this amino group, and this amino group actually acts like, which actually receives the protons. Okay, so we write like this, which actually accepts the protons. So because of this property of the glycine is amphoteric. Okay, so this is the simple thing. Now let us just erase this portion, only this portion. Let us just retain that R group. Sorry, not retain, delete that R group, and let me just put that R group back. And now in this case, if I just erase this R, and all other things, chemicals are actually same here in this thing, COH and amino group, and H is there itself. And let me just put a methyl group. Okay, so methyl group, I just have added the methyl group, and now this amino acid is different, and this amino acid is actually called as alanine. Okay, so this is alanine, second type of amino acid. So now you are getting the point actually. So based on the type of the R group in the amino acid, the amino acid actually changes. Correct? Okay, so different amino acids are possible. Okay, so now let me just erase this CH3, the methyl group, and I just add hydroxymethyl. That is, let me just write CH2OH now. Okay, so hydroxymethyl is added, and now this is a new different amino acid, and this amino acid is actually called as serine. Yeah, so very simple. Now you understood. So based on the different types or like types of R group, we can just classify the amino acids. That's it. Okay, let me erase it and go further. Now let me tell you this alpha amino acids. What are alpha amino acids? Very simple. If you just look into the structure of the amino acid, okay, so we go with a simple structure again. So we have this, and a variable group R group is here, and we have H. Okay, so you could see the four valency groups NH2, COH, and R and H. All these are attached to the common C. This C is nothing but alpha carbon. So all these four valency groups are attached to a single carbon atom. That single carbon atom is called as alpha carbon. Hence this entire amino acid is called as alpha amino acid. Simple. Okay, now let us move further. That is the classification of amino acid based on the number of amino group as well as carboxylic group. Based on the number of amino group and COOH group, the carboxylic group. Okay, here we can classify the amino acid into three types. The first one, that is very common amino acid, that is acidic amino acid. Good example is glutamic acid. For this, I'll give you the structure of this. The second one, okay, so we just go to the second point, that is second amino acid, that is the basic amino acid, and the example for this is lysine. And you go to the third one, that is neutral amino acids, and the good example for this is valine. So this is nothing but the three different types of amino acids based on how many amino groups and how many carboxylic groups are there in the amino acid structure. Okay, we first discuss about the glutamic acid structure so that you can easily understand that. If you look into the structure of the glutamic acid, it's very simple. See, okay, so we just go with the simple amino acid structure, and I write H here, and here the R group. See, you could see the R group now. We have okay, so we have this here. Okay, so this is the simple thing. So this is the structure of the glutamic acid, and you could see how many carboxyl groups are there. Here we have one, and here we have two. So there are two carboxylic groups, and which makes the amino acid acid. So this is acidic, correct? So here we have more number of carboxylic group compared to the amino group. Yes, so that is acidic amino acid. We go to the next one, that is let me erase it because I need space. That is actually the basic amino acid. Now in this case we need more amino group, that is amino group, than carboxyl group, which makes the amino acid basic. That is, good example is lysine. So we just go with the structure of the lysine, that is a bit long structure. So we just go with this, and here I think I'll go with H here, and here we'll just put CH2, 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 and here let us just put the amino group, and in the common structure you could see one more amino group. Okay, so here we have, in the chemical structure of the amino acid, here we have one amino group, and here we have one more. Whereas, look into the carboxyl group, how many we have? Only one. So more amino group compared to the carboxyl group, which makes this amino acid the basic one. Lysine is a very good example. So this is the structure of lysine. Okay, we go to the next one, the neutral amino acid. What is the meaning of neutral here? The number of amino group should be equal to number of carboxyl group. That's it. So that makes the amino acid neutral. So for example, if we just take this structure, we go with the green color actually. So structure, simple structure. So we just put the basic structure. Done. See here. So we have how many amino group? 1, How many carboxyl group? 1. So 1 and 1. So equal. That's it. So which is nothing but the structure of valine, which is a very good example for neutral amino acids. Simple. Hope you got this point. Okay. So now let us again go further. That is actually the classification of amino acid based on the ring structure, that is, call it as aromatic amino acids. Okay, so now further we'll talk about the classification, and let me just give the name for that, that is aromatic amino acids. Okay, so what are these aromatic amino acids? If you just look into the structure of one amino acid, let me just draw the benzene ring here, and let us just put this, and we have this. Okay, so I have to put OH, and here let us put the amino group. Okay, so this is actually aromatic amino acid. This structure is nothing but, let me just give you the example, that is tyrosine. Tyrosine is a type of aromatic amino acid. Why? Because they have aromatic ring here. The benzene ring is attached. Oh, I think I should add OH here. Okay, so this is actually the structure of aromatic amino acid, tyrosine. Okay, simple. Next we go with the second one, that is nothing but phenylalanine. Phenylalanine is actually second type of aromatic amino acid. That means that actually they have this aromatic ring in their chemical structure, that is amino acid is having the aromatic ring, whereas the next one is the tryptophan. Okay, so we just write encapsulate tryptophan is the third aromatic amino acid. Simple. So these are actually the very good examples for aromatic amino acid. Okay, we go to the last concept, that is zwitter ion. So this is very commonly asked questions in the competitive part from amino acid concept, that is zwitter ions. What are these zwitter ions? Okay, so it's very simple actually. At a particular pH. Okay, so at a particular pH, pH, actually the particular the amino group as well as the carboxyl group can be ionizable. So here the amino group as well as the carboxylic group at a particular pH get ionized, that is ionizable. So ionization is possible. Okay, both actually can get charged, okay, at a particular pH. That is actually called as zwitter ion. And let me just tell you one more thing, like during this ionization process in the amino acid, particularly amino acid as well as COOH, the carboxylic group. Okay, so the structure of the amino acid also changes, the structure changes. Okay, so let me just give you a good example for that. Let us just go with the basic structure. Okay, so we have a variable group here, and we have this. Okay, so I should write like this. Okay, next we just see that CH. Okay, and here let us just put the same, and here let us just retain the variable group, and I have to write one more thing here, that is, okay, so we'll retain it, and we just write the amino group. Okay, NH2 here, and you could see here this is actually positively charged, plus is here, and in this case there is no charge, and here you could see at a particular pH. So this is actually at a particular pH, the amino acid is now, particularly the amino group as well as the carboxyl group, is ionized. You could see the charge here, one is minus and one is positive here. Yes, plus. So this here you could see here, this is now negatively charged, but this one is not having any charge, correct? Okay, so at a particular pH, the amino acid can get ionized, particularly the amino group as well as the carboxyl group, both are now charged, correct? As a result, the structure of the amino acid is now changed. It's now quite different, correct? It's changed. Done. So this is actually all about the amino acid. I believe that you understood the entire concept. Thank you very much.